So welcome everyone. We will just wait one more minute to let more people join before we get started. All right, let's get cracking. So welcome everyone to this week's Center for Educational Neuroscience seminar. It is my great pleasure to welcome Mark Haselgrove from the Department of Experimental Psychology at the University of Nottingham. Mark has a background in models of animal learning, but more recently his research has turned to explore learning in humans, and particularly small humans, children. And Mark will be talking to us today about learning about novel and familiar stimuli in children, developmental trajectory. So feel free to put comments into the chat bar, which we can read out at the end of this questions. Otherwise, thanks very much, Mark, and I hand over to you.
Thanks, Mike. I'll just start sharing my screen and getting all the other gubbins going. I can't see you guys now. All I can see is my screen. So can you give me a shout? Can you see my title slide and also a red laser pointer?
Yeah, all looks good.
Yes,
brilliant.
we can.
Okay. Thank you very much indeed. So thank you very much for inviting me today. So yeah, as said, I'm going to be talking today about learning about novel and familiar stimuli in children, a developmental trajectory. And this phenomenon that I'm going to talk about today is called latent inhibition. Conditioning and associative learning has an enormous amount of torturous jargon associated with it, and latent inhibition is one of those pieces of jargon. It's probably a misnomer in many ways. Latent inhibition is neither particularly latent and it's not necessarily inhibition. But it is a remarkably simple behavioral phenomenon. It simply refers to the observation that learning about a novel stimulus proceeds more rapidly than learning about a familiar stimulus. And that's all there is to it. If you take home nothing more than that from this talk, you're doing all right. So latent inhibition was first reported at the end of the 1950s in a study by Lebov and Moore in a conditioning experiment that employed goats and sheep. Here's a goat. And goats and sheep were presented with trials in which visual stimuli were presented. It was the illumination of a lamp or the rotation of some blades, rotating sort of visual stimulus. And each of these were presented separately, and they were paired with a mild shock that was delivered to one of the legs of the sheep and the goats. These two cues were separately established as predictors of shock, and what the investigators were interested in was looking at the extent to which the sheep and the goats elicited a conditioned response, that is to say, a learned response in anticipation of the shock, moving of the foreleg. Ordinarily, learning proceeds quite quickly with this type of procedure, but what Lebov and Moore did is they established one of these two stimuli as familiar to the goats, and they did this by merely pre-exposing the stimuli to the goats. So there would be a session in which, for example, the lamp was simply presented on a number of occasions without consequence, without any outcome. Then once this stage was complete, they would move on to the conditioning stage where the lamp would be paired with shock and the rotating stimulus would be paired with shock. And what they observed was conditioning proceeded more readily, conditioning was more successful to the stimulus that had not been pre-exposed. That is to say, conditioning proceeded well to the novel stimulus compared to the familiar stimulus. And this is latent inhibition. It's demonstrable in many species across the animal kingdom. You can get it in very simple creatures like honeybees, obviously rats and pigeons, but also single cell species. There's some evidence for very simple creatures showing latent inhibition too. There's some evidence for it being present in humans as well. So the top paper by Schnurr and Scher shows the observation of latent inhibition in human eyelid conditioning. So here we have a situation where a cue is established as a reliable predictor of a puff of air being delivered to the cornea of an eye. The puff of air elicits a blink reflex, and with pairings of the cue with the puff of air, the cue comes to acquire the ability to elicit a blink. And this also is normally very successful conditioning. But if you pre-expose the cue without consequence prior to conditioning, then conditioning is less successful. That is to say, you can observe latent inhibition in human eyelid conditioning. Latent inhibition is kind of interesting. It's interesting for a number of reasons. For many years, there's been work looking at the relationship between latent inhibition and schizophrenia. People with acute schizophrenia have typically been shown to not show the latent inhibition effect. So irrespective of whether a stimulus is novel or familiar, people with schizophrenia will typically show comparable learning to these two stimuli. And the effect has also been explored as a possible contributing factor to whether psychological prophylactics may be successful. By this, I mean any kind of intervention that might be used to try and prevent a condition from occurring, some kind of mental health condition. So one example is the acquisition of food aversions after chemotherapy. So one of the psychological side effects of cancer treatment is the acquisition of food aversions. People eat food and then will go and receive chemotherapy, they feel ill, and they will acquire a food aversion. So one technique for trying to reduce the food aversions that people ordinarily experience to their everyday diet is to use what's called a scapegoat food. This would be a food that patients would eat just before their treatment, and the scapegoat food takes the blame for the association with illness. That reduces the number of food aversions that people have to their normal diet. So it seems to work quite well as a scapegoat, but it works best if that food is novel. Okay, if a familiar food is used as a scapegoat, then that effect works less well. And the analysis goes that the scapegoat food forms an association with illness in a conditioning type way, and this works particularly well with novel food compared to familiar food. That is to say, you get latent inhibition. And it's also been suggested as a possible way of trying to alleviate some of the anxiety that might be associated with certain phobias as well. So participants who experience traumatic events at dentists are more likely to report the acquisition of a dental phobia if they've never been to the dentist before that traumatic event. If participants have experience going to the dentist prior to that traumatic event with no consequence, then they are less likely to acquire dental phobia, which looks like a latent inhibition effect. The dentists is more likely to be associated with the traumatic event if they're novel compared to if they're familiar. There are probably about as many theories of latent inhibition as there are people investigating latent inhibition. But generally speaking, there are two clumps of theory that permit us to explain it. On the one hand, there are attentional theories, and these suggest that pre-exposing a stimulus, that is to say, making it familiar through mere exposure, reduces the amount of attention that is paid to that stimulus. And that has a consequence of subsequently slowing learning to it. You're more likely to learn about a stimulus if you're attending to it. A second class of theories is so-called proactive interference theories. And the idea here is that conditioning is attenuated to a stimulus which is familiar because that pre-exposure stage has a proactive interference effect on it. When you're trying to learn, for example, that a cue signals an outcome, that's going to be less successful if you've got a bunch of memories that precede that where the cue signals the absence of the outcome. Okay. So that's latent inhibition. That's why it's kind of interesting. One interesting sort of note about latent inhibition is that it's pretty easy to get in animals. It doesn't take much effort to observe latent inhibition in the study of conditioning in animals, but it's a little bit more difficult to observe latent inhibition in human participants. For many years, it's been thought that in order to observe latent inhibition in humans, one needs to embed the pre-exposure stage where the stimulus is made familiar in the context of a so-called masking or concurrent task, where participants are engaged in some other sort of task, and the pre-exposure happens kind of incidentally to that. And this sort of led people to think about latent inhibition in the sort of context of recapitulation theories, and the interest started to develop in the developmental psychology of latent inhibition. And the first part of my talk, I'm going to summarize 4 studies that have looked at latent inhibition in children, and then as I go, talk about some of the limitations of these studies, and then talk about a more recent study that we've done. So one of the most influential studies of latent inhibition in children was reported by Kanyal and Lebov in 1986. They recruited 240 children and split them into age groups that you can see in the top right-hand corner. And in their task, they had a pre-exposure stage in which participants were pre-exposed to a black square, for example. This is the pre-exposed stimulus in the middle. I said a moment ago that often, in order to study latent inhibition, the pre-exposure is embedded within a concurrent task. And the concurrent task that children were engaged with was pressing a key underneath a picture of a plant, but not an animal. And these pictures were flanked the pre-exposed stimulus. So you'd have an animal sometimes on the left and sometimes on the right, same with the plants. And what participants had to do is simply press the space bar, the button that was underneath the plant. But the manipulation of an interest was the pre-exposure to the black square, because in a subsequent second stage of the experiment, participants now had to learn through trial and error that they would receive reward whenever they pressed the same button when the black square was presented. So they were presented with the same apparatus, but now the pre-exposed stimulus, here the black square, would be presented on one side of the apparatus, and a novel stimulus, here a white square, would be presented on the other side. And participants had to press the button in order to receive reward. For young children, I think this was marbles. For older children, this was verbal reward. So here, this particular group of participants, for example, have to press the button underneath the pre-exposed stimulus. Another group of children, for example, would have to press the button underneath the novel stimulus. And the dependent variable that they measured was the number of training trials required to reach a particular criterion. In this experiment, the criterion was 10 consecutive correct responses. So in the graphs I'm going to show you in just a moment, larger numbers mean poor learning. So there are 2 experiments in Kanyal and Lebov's paper. Here's experiment 1 on the left and experiment 2 on the right. They're pretty much doing the same kind of thing. On the y-axis of both graphs, we've got mean trials to criterion. So remember, again, bigger numbers is poor learning. And on the x-axis of both of these graphs is the age of the children. And these lines at the top here, these are the data from participants in the groups that were pre-exposed to the stimulus that was paired with the reward. These lines down here, these are participants who are in the group for whom the novel stimulus was paired with reward. And what I hope you can see is the presence of a latent inhibition effect in children who are relatively young, but not in children who are relatively older. And this effect is also reproduced in their experiment 2. So that looks like there's, at face value, a peculiarity of the developmental trajectory of latent inhibition, in that it's present in younger children, but it goes away in older children. There are, however, a few issues that I have with this particular study. First of all, statistically, the crucial age group by stimulus interaction for experiment 1 is certainly significant. You know, they report it as p equals 0.05. When you actually push the t value, the f ratio through some numbers, it is only just significant, which is fine. You know, just significant is still significant. But their experiment 2, which is reported as significant, p is less than 0.05. Actually, when you look at the f ratio, it's not significant, which is frustrating. It's not devastating, but it's quite frustrating. More frustrating for me is the possibility that what is being explored in this particular study is not a developmental trajectory of some kind of interesting cognitive phenomenon, but in fact just a sort of basically a ceiling effect, because the absence of latent inhibition that we see in the older children is being driven pretty much by the fact that the older children find this task trivially easily. The criterion for the task, mean trials criterion, was 10 trials, and the older children are getting this task in 15 trials. So out of only 5 learning trials, they're then making 10 consecutive trials. Evidence that supports this, when the investigators omit the very oldest groups in these 2 experiments, then this significant stimulus by age interaction falls apart. So there's a big caveat to this study that what might appear to be the presence of latent inhibition in younger children, but not older children, may be a consequence of a performance scaling effect. Another issue that was raised more recently by McLaren et al. In a paper published a couple of years ago is that the conditions of pre-exposure might not necessarily permit our effect to be explicable just in terms of mere exposure, but instead in terms of some other kind of psychological phenomenon. So, for example, during the pre-exposure stage of the experiment conducted by Kanyelin and Bov, participants are pre-exposed to this black square. That is certainly the case. But the concurrent task involves a discrimination between animals and plants. Which of course means that the pre-exposed stimulus is irrelevant to this task. So perhaps one reason why we might not see particularly good learning to this stimulus is because it's been established as task irrelevant rather than it being exposed. So recently they've sought to try and reproduce this phenomenon with a task that is less susceptible to this problem. So in this task, participants are in stage 1 required again to discriminate between 2 task relevant stimuli. This is children are presented first with a fixation point, then there is a warning signal, and then they have to press one of 2 keys depending on whether a dinosaur or an animal is presented, and whether or not they get that right depends upon the feedback that they get. So if they press the right key and it's a dinosaur, they get yeehaw played. The important part about this pre-exposure is that the pre-exposed stimulus is this warning signal here. Okay, so it's not an imperative stimulus in that participants have to respond to it, but it does. Participants are assumed to attend to this stimulus by virtue of the fact that it warns participants that an imperative stimulus is coming up, a dinosaur and an animal. Once this pre-exposure stage has been complete, they move on to a second stage of the experiment, which is a go-no-go task, where again there's a fixation point, and then either the pre-exposed stimulus or a novel stimulus is presented, and participants have to learn either to press the space bar, i.e. a go trial, or withhold their response, a no-go trial. So there are 4 stimuli in stage 2. There are 2 novel stimuli, and one of these novel stimuli participants have to respond to, go. Another stimulus they must not respond to, no go. And then there are 2 familiar stimuli as well that have been pre-exposed, and these are also go and no-go stimuli. So here would be an example of a no-go trial with a novel stimulus. They recruited just over 100 participants, and they split them into the age groups that you can see there. They also included some undergraduates in the study as well. Let's have a look at some data. So here are some data from the GO trials during stage 2, where we're looking at the responding on the spacebar during the pre-exposed and the novel stimulus. On the y-axis of this experiment, what we've got here is mean response accuracy %. So now bigger number means better learning. What I hope you can see is we can see the presence of latent inhibition in the youngest children. So the grey bars is the mean response accuracy to the novel stimulus, and the darker grey bars on the left of each pair is the pre-exposed stimulus. So learning is more successful to the novel stimulus compared to the pre-exposed stimulus in our youngest children. This effect starts to weaken, however, as we go up the age groups. When we get 10 to 11-year-olds and then undergraduates, the effect is abolished, which is, at face value, a reproduction of the kind of results that was shown by Kanyel and Lebov, the presence of latent inhibition in young children between the ages of 4 and 5, but not older children. A problem with this study, however, is that this effect was only observable during the GO trials. If you look at the no-GO trials, then if anything, we see the opposite pattern of results. Now, we see more successful learning about the pre-exposed stimulus than the novel stimulus, at least numerically. And there's no evidence that this effect reduces in magnitude as the children age. Which is a peculiarity until you have a little think about the conditions of pre-exposure, which was raised by the authors themselves. They suggest that one reason why the effect is present on the GO trials but not the no-GO trials is because of a general response inhibition effect that was conditioned during the pre-exposure stage. So if you recall, during stage 1, participants were required to press one of 2 keys when they saw a dinosaur and an animal, and the pre-exposed stimulus was established as a warning signal for these imperative stimuli. Here are the instructions on the right. They said to children, before you see them, there will be a signal to warn you that the animal or dinosaur is coming. Remember, as soon as you see a dinosaur, press the X key. As soon as you see an animal that isn't a dinosaur, press the dot key. So it's possible that the reason why children were responding quite a lot to the novel stimulus, but not the pre-exposed stimulus on the GO trials, was because they'd learned to not respond during the warning signal, some kind of general response inhibition effect. And that would produce what appears to be latent inhibition on the GO trials. And it would also provide a reasonably natural explanation for why the opposite pattern of results emerges on the no-GO trials. General response inhibition can also explain another another demonstration of latent inhibition in children that was reported by Lebov and colleagues in 1976. So in this experiment, children around the age of 5 were sat in front of a table and asked to learn the objects under which a particular marble was hidden. And what they were interested in measuring was response time to pick up the correct object for marble reward. On the y-axis of this graph here, we've got mean reaction time to pick up the correct object when that object had been pre-exposed. That is to say, the objects had been pre-exposed to the children with the absence of the marble, and when the objects were novel. These different coloured bars just represent different sort of counterbalancing groups. But the overall effect is that reaction times was faster to the novel stimuli compared to when they were pre-exposed. And the reason why response inhibition might explain this particular effect rather than something more akin to mere exposure is the nature of the instructions that the children were given again. So on the right, you can see that they were told that when I raise the screen, you will see a table and several objects on the table. Please do not touch them. Sit with your hands crossed or by your side as you like. I want to measure with this watch how long you can sit quietly watching the table and the things on it without saying or doing anything. So it's perhaps not unreasonable to suspect that this instruction led to children not reaching out to reach for objects in the pre-exposed condition, and that might have led to their reaction times being slowed. It's also the case that all these children in this particular experiment were in kindergarten, so it might have been the fact that we would have seen a similar effect in children who are greater than 5 years old. We don't know, unfortunately. The final experiment that I want to tell you about before I move on to our data was again conducted by Lebov's laboratory here in 82. In this particular experiment, children were presented with circular stimuli projected on a wall, gloriously old school, and these 2 stimuli differed. They were always circles, so there'd be one circle on the left, one circle on the right, but they would differ along a number of dimensions. They would differ in terms of their overall size, their color, and the number of circles within them, and the color of those circles within them as well. And for one group of children, they were told to just look at the stimuli during the pre-exposure stage. A second group of children were engaged in a concurrent or masking task during the pre-exposure stage. They were presented with 3 Hebrew letters between the circles, and the children were asked to unscramble those letters to make a high-frequency noun. And a third group of children didn't receive any pre-exposure during stage 1. In the second stage, the children were just asked to point at whichever circle they thought was right, and they were given verbal feedback. And they were actually always the actual contingency here was that the big outer circle was always the correct one. These were 10 to 11-year-olds. I couldn't actually ascertain from the paper how many were in each group, but they're all around 10 years of old. And the dependent variable was the mean number of trials to criterion. So again, bigger numbers refer to poorer learning. And here are the data. So on the y-axis, we've got these mean trials to criterion. This bar on the far right, these are the children for whom the stimuli were novel during stage 2, the actual sort of interesting learning stage. And we can see that their trials to criterion were relatively low. In children for whom they'd been pre-exposed to these stimuli with a concurrent task, a masking task, we can see that learning was slower. There were more trials to criterion. Interestingly, there seems to be some kind of effect of the concurrent task. When there was no concurrent task during pre-exposure, then latent inhibition was not observed. Now we see relatively rapid learning. That is to say, at least as rapid learning as the group of children for whom there was no pre-exposure. Okay, so as an interim summary, there's at least some converging evidence for latent inhibition being present in young children, but not in old children. As I've noted, there are a number of shortcomings that both the authors and other people have noted with some of these tasks. But I also think it's a bit weird because if we think of latent inhibition as a task that in some way is a function of organisms' ability to increase or decrease the amount of attention that they're paying to a stimulus, then we might put it squarely in the camp of tasks that are measuring executive function. And we might expect to see an improved developmental trajectory for an executive task between the ages of, say, 4 and 14, rather than the presence of the effect in younger children and then the lack of the task later on. So this sparked our interest, and we wanted to look at it ourselves. We wanted to look at it ourselves with a task that was not so easy that older children would hit a performance ceiling. We wanted to look at it with a task that doesn't make the pre-exposed stimulus task irrelevant, so they're just learning to ignore the task at the pre-exposed stimulus because it's got nothing to do with what they're doing during pre-exposure. And if possible, we'd like a task that's within subject, so that for each individual child, we can get a measure of learning to a pre-exposed stimulus, a familiar stimulus, and also a measure of performance to a novel stimulus, and use that as some kind of measure of our latent inhibition effect. So that's what we did. We ran an experiment at the University of Nottingham Summer Scientist Week. This is a week-long public engagement event that's held at the University of Nottingham. We recruit 4 to 17-year-olds and also their parents. They come into the university every day for a week. And they participate in experiments, and as a consequence of participating in the experiment, they receive one of these little tokens, which they can then use to spend on one of lots of exciting games in a large area away from the experimental labs. And it's great. It's a really nice engagement and event in and of itself, but it also permits us to test quite a lot of children within a week. We ran an experiment this year at Summer Scientist Week. I only had one experimenter running an eye tracking experiment, and I managed to get around 50 children's worth of usable data within 5 days, which was really really nice. If you're interested in being involved in Summer Scientist Week with us, then feel free to give me a shout and I can put you in touch with the organisers. So this was run, this experiment I'm going to tell you about was run at this public engagement event. we needed to decide how many children we needed to run. So we looked at the most recent study of latent inhibition in young children. This was the 2021 paper by McLaren and colleagues. They reported partial letter squared values of around 0.07 and 0.8 for the interaction, the crucial interaction that involved age group and stimulus. We were quite keen to run regression for our analysis, however. We were of the opinion that age is a continuous variable, so treat it as such rather than clumping children into age groups. So we converted it to F squared, their partial letter squareds. It's always a pleasure to read Cohen's book. And G Power told us that we probably need somewhere in the region of 154 or 178 children for regression with one predictor that was two-tailed. So over 3 years of Summer Scientist Weeks, we managed to recruit 175 children, just under 90 females, just under 80 males, who had a mean age of 8.5. Just because we were glutton for punishment, we also recruited 175 adults as well as a sort of comparison group, just to sort of contextualize performance in the children. As you can imagine, these were undergraduates, psychology undergraduates, hence this sort of skewed ratio of gender, of sex, and relatively young age. We have been running latent inhibition studies more recently using Prolific, where you can say, I'd like a matched sample and also I'd like a nice broad age range, and our effects are reproducible in a much more representative sample. So, and the task that we used to investigate latent inhibition is one that we've used and other laboratories have used in recent years. It's a letter prediction task. We've used it to look at the relationship between latent inhibition and schizotypy on the basis of studies that have looked at latent inhibition and schizophrenia. But the task in and of itself is quite useful for investigating latent inhibition in a number of contexts. And I'll spend just a little bit of time going through the task so that you can get a bit of an understanding of what we're measuring and what we're looking at. So like in previous studies of latent inhibition, there's always two stages. There's a pre-exposure stage, which is used to establish a stimulus as familiar. And then there's a test stage where we're looking at learning to that familiar stimulus compared to a novel stimulus. And during the pre-exposure stage, what participants see on screen is a succession of letters appearing. Each letter appears on the screen for a second, then there's a shortened stimulus interval, then there's another letter. Okay, and during the pre-exposure stage, this happens relatively quick. I mean, I think the pre-exposure stage only lasts for about 3 or 4 minutes. Participants are asked to just watch the sequence of letters appearing on screen, and they are just instructed to say out loud the letters as they appear on the screen. Okay, so for example, it might be, and within this pre-exposure stage, there are letters D, M, T, V, and crucially, our pre-exposed stimulus, which in this particular case was letter S. So S would come on, interval D, M, T, V, S, and participants would just be saying the stimulus out loud. Once this stage was complete, participants moved on to a second stage where, again, they would see this sort of succession of stimuli letters appearing on screen, each one for 1 second separated by a shortened stimulus interval. But now they were given a new task. They were told now to watch the sequence of letters appearing on the screen, and what we wanted them to do is to try and predict when a letter X was going to appear. And we told participants if they thought they knew when the letter X would appear, then they could press the space bar early in the sequence. That is to say that they could press the space bar before the X appeared. If they couldn't do it, then they can just hit the space bar whenever X appeared. And embedded within this sort of sequence of letters was a training structure. So we had trials in which letter S preceded letter X 20 times, and we had trials in which letter H preceded letter X 20 times. So here we have trials with the pre-exposed stimulus predicting X, and we have trials in which a novel letter H is preceding letter X. And these are all embedded within lots and lots of filler trials, so lots and lots of exposures to these filler stimuli D, M, T, and V, which are also present during pre-exposure. So these are relatively infrequent trials. And there's also a bunch of sort of filler trials in which D, M, T, and V predict X as well. So the contingency between S and X and H and X is not perfect. There are trials in which the target stimulus appears, but it's not predictable. But there are a whole bunch of trials where it is. S, whenever it's presented, is always followed by X, and H, whenever it is presented, is always followed by X. The question of interest is whether response times to the target change as a function of whether or not it's preceded by S or H. We might imagine that participants will come to anticipate X by virtue of this consistent relationship between S and H and the target. So response times will start to diminish as training progresses. But the question of interest is whether that reduction in response times will be more slow to the pre-exposed stimulus S than the novel stimulus H. And that's certainly what we observe in humans, in adult humans, sorry. So on the y-axis here, we've got mean reaction time over 10 two-trial blocks. And here we've got a reduction in response times here to the novel stimulus, the non-pre-exposed stimulus coming down. And quite stubbornly, response times in this particular experiment were not coming down to the pre-exposed stimulus. We rarely see a total absence of a reduction in response times to the pre-exposed stimulus. Here's another example of exactly the same experiment conducted, but it was done online. Now we see a reduction in response times to both the pre-exposed and the non-pre-exposed stimulus. The non-pre-exposed stimulus is shown with the dotted lines, and the pre-exposed stimulus is shown in the solid lines. And we can see what looks like a learning effect. We have no difference in response times at the outset of training, and a difference emerges over the 10 two-trial blocks. But this rate of learning seems to be more rapid to the novel stimulus relative to the familiar stimulus. And we take this as a possible measure of latent inhibition. And the question of interest for us is, if we provide this task to children, what will we see? Will we see the kind of developmental trajectory that other people have seen, that is to say, latent inhibition in young children, but not old children? So this task was given to children. Obviously, we changed the language a little bit, so we replaced the long words with shorter words. The instructions were presented verbally. We didn't use words like experiment. This was a game that they were playing, and children engaged well. Some initial data processing was conducted. So we removed trials with response times that were very, very long or very short. That resulted in a little bit of loss of data from the adult and children's data. Any participants who failed to respond on half or more trials were also removed. That resulted in 8 children and 3 adults being removed. And any missing values, that is to say, for example, a trial where they simply missed responding to X, were replaced with imputation using missed forest. And that counted for around between 14 and 60% of trials. I'll talk more about that later on, though. So the first 2 graphs I want to show you, just characterizing the rate of the response times to the pre-exposed and novel stimulus to the children, which is shown in the left panel, and the adults to the right. So once again, on the y-axis of these graphs, we've got mean response time over 10 two-trial blocks of training. So these are 20 occasions in which S is paired with X and H is paired with X. And in the children, what I hope you can see is that response times reduce more rapidly to the novel stimulus compared to the pre-exposed stimulus over training, which looks like a latent inhibition effect. We see the same pattern of results in the adult data as well. The stats essentially show that adults are responding more quickly than children, which is no surprise really, and that also there was a stimulus by trial block interaction, so that the difference in response times was significant from trial 2 onwards in both the children and the adult data. So that just provides a sort of overall characterization of what this sort of response time looks like as training progresses. More interesting, however, is to ask the question, does this effect vary with age in the group of 175 children? So get an initial feel for this, we performed analysis of covariance, where we did a 2x10 ANOVA, so 2 being stimulus, pre-exposed or novel, 10 being 2-trial block, with mean-centered age enters as the covariate. And what we find is we get a significant interaction between the covariate and both our effective stimulus and also the stimulus by trial block interaction. So depending on how old the children are, something different's happening between the pre-exposed and the novel stimulus, which is a first indication that we've got some kind of possible developmental trajectory going on. In order to try and characterize this, we computed one score that gives us a measure of latent inhibition. We took overall, over the entire stage 2 of training, during the test stage, we took the mean reaction times for the pre-exposed stimulus, excuse me, the mean over the 20 pre-exposed stimuli, and 20 trials for the novel stimulus during the test stage. And then computed a latent inhibition score. So we just took the response time overall for the pre-exposed stimulus and divided that by the sum of the response times to the pre-exposed stimulus and the novel stimulus. And with this measure, any score that's greater than 0.05 indicates latent inhibition. That is to say, faster responses to the novel stimulus compared to the pre-exposed stimulus. And a score of 0.5 would indicate no difference in response times to those stimuli, so no latent inhibition. And the developmental trajectory looks like this. So on the y-axis here, we've got our latent inhibition score that I've just described to you. And on the x-axis, we've got the children's age from 4 to 15. And we ran, what we've got on this is this dotted line that runs across here. This is this sort of 0.5 part on the scale where there's no latent inhibition. This solid black line is our regression slope, and these dotted lines that surround it, these are 95% confidence intervals around the regression. And the regression was significant. So we had an R squared of 0.073, which is significant. And the nature of this regression indicates that as children get older, the magnitude of latent inhibition gets larger, rather than, as the earlier literature would suggest, the opposite direction. Earlier experiments had indicated the presence of latent inhibition in younger children, but not older children. So if anything, rather than seeing a slope like that, we would expect to see a slope like that, but we don't. One concern that we had was that we omitted children who had very, very fast or very, very slow response times. And we also conducted imputation where we guessed, mathematically, missing data. And we were a bit worried that we did that because we thought, well, it may not necessarily be the case that missing data was evenly distributed across age. It may be the case, and probably, and certainly is the case, that younger children have greater number of misses than older children. So perhaps we're just making up more data for the younger children than we are the older children. So we reproduced this regression. We didn't do any fiendishness. No, we didn't remove any outliers. We didn't do any imputation. We included all children, and the regressions remained. We still get a significant relationship between age is still a significant predictor of this latent inhibition score. And one thing that we were quite interested in looking at is the precise, some indication of when in the development of children does latent inhibition emerge. And because we have this point, this sort of 0.5 point on the y-axis, which indicates essentially chance, where there's no difference in response time to the familiar and the novel stimulus, we can use this to ask the question: at what point does the lower bound of the 95% confidence interval cross this chance level, which might then give us some kind of indication of the age at which latent inhibition emerges. And it's around here. The lower bound of the 95% confidence interval crosses chance at a point when the children are between 7 and 8. It's 6.7 years if people are very, very excited by precise ages, which was kind of interesting for us for a variety of reasons. We did some further analysis. We were kind of interested in looking at whether or not this relationship was best characterized by a linear regression. We also ran a number of other forms of regressions as well, and we found that quadratic and cubic regressions were ever so slightly bigger. Their R squares were better. We conducted extra sums of squares test to see whether the F ratio of the sums of squares over the increase in degrees of freedom these more complex regressions afford you improved stuff. They didn't. So it looked like the linear regression provides a sufficient explanation for the relationship between age and this latent inhibition score. One thing that we were also concerned about here, if you cast your mind back to the start of the talk, where I described the first experiment by Kanyal and Labov, where they observed latent inhibition in young children, but not in old children. The problem associated with that particular study, as I described it, was a performance ceiling effect. The reason why they didn't see latent inhibition was because the children found it too easy. One concern that we had is that the younger children just found it too hard, and that they can't name letters, or they're scared of the experiment, or whatever it is. We were worried that the very young children found our task too hard, rather than the old children finding it too easy. We weren't too concerned that the older children find it too easy, because we knew that our latent inhibition task was reproducible in adults. So one thing we thought would be useful to do was to truncate our sample at the age at which the children start to show latent inhibition. So this point at which the 95% confidence interval crosses chance, we can say that, well, let's truncate the sample at that point, remove all children from that younger point age there, who on average don't show latent inhibition, and then just rerun the regression on the older children. So then we could maybe make a more conservative conclusion. We could say, in a sample of children who on average all show latent inhibition, do we still see that age is a predictor of this latent inhibition score? And then the answer is yes, it does. Even with this sort of truncated sample, we still see a significant regression. Latent inhibition still increases in magnitude as the children get older. Okay, so in summary, children, like adults and animals, maybe, learn faster about novel stimuli relative to familiar stimuli. This effect, latent inhibition, gets bigger with age and looks like it's linear. It's also pretty resilient. We can get this effect without doing fiendish things like removing outliers. We can truncate our sample where we remove really quite young children, and we still see an improvement in this effect with age. I haven't shown other analyses here because of time reasons, but the effect isn't dependent on our particular measure. An editor who looked at this paper recently said, well, maybe the reason why you're getting this effect is because you're using the ratio. So we've recomputed the latent inhibition score. We calculate different scores, just subtracting the response time from one stimulus from another. Do we still get a significant regression? Yes, we do. And we've also separated it in terms of responses that were performed before the target, and looking at, do children who are young and old also perform differently in terms of just anticipatory responses? And the answer is also yes there as well. So it seems to be quite a resilient effect, which we're quite happy with. But it is contrary to the existing literature, which, if anything, shows the presence of latent inhibition in younger children, but not older children. So that begs the question of why, the most dangerous question in the world to ask. And I don't have a particularly good answer, unfortunately, so apologies. I mean, Kanyal and Labov, their explanation for why latent inhibition emerges is an attentional one. They suggest that every time you pre-expose a stimulus, you get a reduction in attention to that stimulus, and hence slow learning about it. And that's what they say happens in animals and young children. If you were to pair that stimulus with an outcome, like you would do in a normal conditioning experiment, then the opposite happens. You increase attention to a stimulus. And their analysis of this effect, that the presence of latent inhibition in young children and the absence in older children, they attribute to young children merely experiencing the pre-exposed stimulus, whereas older children start to make a response of some form to the pre-exposed stimulus. So they may either sub-vocalize it or even vocalize it. So they might just sort of say, there it is, for example, whenever the stimulus appears. Younger children and animals, they assume, do not do this, and therefore they show this latent inhibition effect because the stimulus is never paired with an outcome. But in the older children who are learning to say something, for example, or sub-vocalize something to the stimulus, they don't show this latent inhibition effect because it is paired with something. That particular analysis isn't particularly amenable to our data because we actually make children say something to the stimulus during the pre-exposure stage. We were very keen to not have this pre-exposed stimulus irrelevant to the task in stage one. We wanted it, if anything, to be task relevant. We wanted them to say all the stimuli during stage one, so they were engaging with it. So if anything, they shouldn't show latent inhibition at all, but of course they do. So it's not that. And another possible explanation to consider when looking at the different kind of effects that we see in different laboratories is to note that different laboratories use different sorts of concurrent tasks during pre-exposure. So Canulin and Bob say that all subhuman vertebrate species that have been examined for the effects do exhibit latent inhibition, but there's a curious discontinuity amongst human subjects. Young children show the effect, older children and adults do not, and they clarify that with except under special masking conditions. They don't say what those special masking conditions are. In a recent review of the literature, Byram, Setfi, and Murphy attempted to categorize the different types of masking or concurrent tasks that were performed during the pre-exposure stage of latent inhibition studies in humans, and you can classify them in terms of whether the familiar stimulus was established as task irrelevant during pre-exposure, or whether the familiar stimulus was established as task relevant during pre-exposure. And maybe ask the question, well, is it the case that this discriminates between whether latent inhibition is going to be either absent or present in older children? Unfortunately, it doesn't. We can see both the presence and the absence of latent inhibition in older children in both of these two sort of types of pre-exposure stimulus, pre-exposure task. So I think I'm going to leave the last word today to Michael. This is a great paper. I really, really like it. And one statement that stayed with me is shown at the bottom. It says, one of the biggest current challenges is to calibrate measurement systems to afford age-level sensitivity while at the same time retaining conceptual continuity over large spans of time. That is to say, still constituting measures of the same process at different ages. And that's my concern with these studies, is that we haven't got a particularly good handle yet on whether we're measuring something that is of comparable difficulty to older children and younger children, and therefore obscuring the presence of what might be a real developmental trajectory with differences in simple task difficulty, for example. There were clearly effects that were consistent with that analysis in early studies, and I think our very youngest children did struggle to find the task, did struggle to use the task in the children who are 4 and 5. And we went to some lengths to try and omit children that did struggle with the task. But I still worry about this particular problem of calibration. So it may be the case that there were multiple developmental trajectories out there, and sometimes it looks like latent inhibition gets better with age or it attenuates with age, and it's just down to the difficulty of the task depending on your age. So the reason why I've called this a developmental trajectory rather than developmental trajectory sort of acknowledges this problem of scaling effects in older and younger children. All it remains for me to sort of say now is just to sort of thank the people who actually conducted the experiment on 175 children, which is no mean feat at all. Emily Gray and Sabina Boganovic were both undergraduate students at the time, and they tirelessly worked with all the children. And these experiments all comprise part of Sue Lynn Ma's doctoral thesis. Sue's now working at PsychoPy, and she helps us program all the experiments that we've done. And it also remains for me to say thank you, everybody, for your attention today and listening to my talk. So thank you very much indeed.
Superb. Thank you so much, Mark. Really fascinating stuff. I'd love to get into the nitty gritty of learning and the nuts and bolts. So if anyone has any questions, do stick them in the chat bar. If you do that, you will save Mark from me asking a series of tough
nitty gritty questions.
So yeah, feel free to put your hand up if or put a question in the chat bar. So yes, Mark, I'm not trying to decide whether to ask a really kind of fine-grained question or a big one. So maybe I'll do both, right? So that here's the
both is fine, Michael.
Fine-grained question. If you think that learning depends on the statistics about how well the cue predicts the outcome, and you average over the familiarization phase and the task phase.
Oh, sorry, it was averaged just across the task phase, not the familiarization phase. Sorry, Michael.
Yeah, no. So I'm just saying, the brain doesn't know that, right?
Okay, sure. Yeah, fair enough. Yeah.
So it doesn't know about the different phases, and the familiar. If you average over all the phases, the familiar cue is just less predictive of the outcome.
Yeah. Yeah.
Because previously nothing was happening, right? So could you explain it just on a statistical basis?
Yeah, absolutely. Yeah. So when I said that early on, there are as many explanations of latent inhibition as there are people interested in latent inhibition. Michael, you are now a person who are interested in latent inhibition because you have a theory of latent inhibition. You're not the first person to come up with that one. Yeah. The contingency is degraded between cue and outcome in the pre-exposure phase. So that is one explanation as well. One interesting feature of latent inhibition is that it doesn't just degrade the contingency of it doesn't just attenuate the acquisition of excitatory learning, it also attenuates the acquisition of inhibitory learning as well. So if you're trying to learn that a cue signals the absence of an outcome, if you pre-expose that cue, it still attenuates that acquisition of inhibitory learning. Now, I think you might expect it to be even more inhibitory with pre-exposure rather than the opposite. So that has been taken to be some evidence, but that alternative sort of looking at the attenuation of condition inhibition is far less well studied than the other way. But yeah, that's another explanation for it.
Great, thanks. And yes, sorry, Joe, I stole your question there. Joe Taylor also has a follow-up. I guess I could ask you to reflect on the different stroke similarity between latent inhibition and other forms of statistical learning. Is it in some way a special mechanism underpinned by specific neural mechanisms?
Yeah, it's a great question. I think I could answer that yes and no, actually, depending on what mood I'm in, to be honest. Sometimes I think it's just a general process, or it's a phenomenon that reflects a general process under either associative or statistical learning. I don't have a particular. Well, I do actually have a line in the fire. I think it's associative learning, but I'm quite happy for it to be statistical learning as well, ish. And I think it's a general process because we see it across so many species and across so many different tasks. So on the one hand, do I think it's special? No. On the other hand, I think it's multiply determined as well. I think because there are so many different explanations for latent inhibition, and there are studies which suggest that, yeah, it does look attentional, but it also looks like it's a contingency thing. Oh, it also looks like it's proactive interference as well. That's bound to interact with whether or not it's special or not. So yes and no. That's a really rubbish answer, isn't it? I'm sorry, Joe.
Covers all the bases, though, doesn't it?
Yeah. Safe.
All right. Let me ask you the kind of step back question, which for our interest in educational neuroscience is how teachers should use novelty in the classroom. Yeah, so that they're kind of dealing with all these little brains out there, and the hippocampuses in all those little brains really like novelty, and you lay down episodic memories much better for novel stimuli. But if you want to do conceptual learning, those little brains don't like novelty because that's harder to fit into schema. You want schema consistent knowledge gets integrated better, and stuff that doesn't seem to fit with your prior knowledge is much harder to learn. So where would you see novelty playing the role for teachers? How are they best?
What recommendations would I give a teacher, Craig? My wife's a teacher. I will just I haven't run
What
this
do you
past
say to her?
I haven't run this past my wife yet, so dangerous territory immediately. Yeah, it's great. I mean, I can actually reverse latent inhibition. I can actually get participants to learn more rapidly about a familiar stimulus than a novel stimulus. It all depends on what the context that they're being presented within is like. If the context itself sustains novelty or sustains familiarity, then you can push things around that like that. I hadn't thought about it in terms of conceptual learning quite so much. So that's something I'll have to sort of dwell on a little bit. One place that I had thought that it might be kind of useful, and I've been chatting with a couple of people about this, is in terms of how it's been used in the past as a prophylactic to try and avoid the acquisition of certain cases of fear. And one thing that we're certainly finding at university level is becoming more prevalent is fear of failure. Students really do feel quite averse to summative assessment. They're quite a lot of fear of being given feedback. And of course, one way in which we can try and look at the way in which we can ameliorate that fear of failure is to give formative assessment to students prior to summative assessment. That sounds an awful lot like a latent inhibition study. So you're giving students the opportunity to experience the stimuli associated with assessment without the outcome, that is to say, the feedback. And under those circumstances, we might imagine the fear of failure to be attenuated somewhat. And that's, I mean, it's not just a sort of conceptual redefinition. You know, we do know a reasonable amount about the conditions under which latent inhibition is really well demonstrated, and the circumstances in which we can break it. So, you know, if certain things like that in the classroom are amenable to explanation in terms of latent inhibition, then we can make recommendations in terms of when it might not help. But in terms of conceptual learning, that's a great thought. I hadn't thought about it in that context. So that's really interesting.
Super. That's a fantastic, thought-provoking answer. I have one final question. I was looking at your graph and trying to judge across different ages whether there was a normal distribution in latent inhibition. And really just to move into an individual differences framework about do kids differ in or of any age in how much they are attenuating attention in response to cues, and what would underlie individual differences on this particular variable?
So I got chatting about this recently. So one thing I have been looking at is the extent to which there are differences in variability depending on the age of the child. Someone suggested surely you're going to see a lot more variability in response time in the young children than the old children. And that bugged me. So I looked at that yesterday, actually, in response to someone's question. And I was actually quite surprised. We don't see a change in response time variability. If you just track the standard deviation of all the trials and plot that as a function of age, that's not a predictor. In terms of what the underlying mechanism is at the moment, I don't know. Every time I talk to Lucy Cragg about this, she seems to think that it's got something to do with task switching or shifting attention away from one stimulus and to another, maybe from away from the context in the background towards the stimuli of interest, or maybe it's something shifting away from a sort of reactive response to the target to an anticipatory response to the cue instead. And she keeps on saying to me, look, that happens between 6 and 7, Mark. That's exactly what I'd expect. So maybe I think Lucy might be right. She normally is.
Okay, are there any other questions or comments out there? Otherwise, we are past 5 o'clock, so it just remains me to thank Mark again for a fantastic presentation. Many thanks, Mark, and
thanks very much for hosting me.
Thanks, everyone.